Sickle cell anemia also we have discussed. Now
Yes,
see,
ma'am.
if you remember in yesterday's class, I told you that there is a mutated gene for color blindness. See, the X to the power C, I told you this is the mutated gene, whereas the normal X chromosome is there, right? Which is not mutated, it's like the normal one. But the one that gets mutated by a chance, we call it as the mutated gene, and this XC causes color blindness. Clear?
Yes.
So XCY is the affected male, XC is the affected female, and then the XC normal X is the carrier female, right? This is what we discussed.
Yes.
Okay. Now, see these are the possible crosses that could be there, like a normal woman with a colorblind man. These are the possible crosses. It could also be a carrier woman alongside a colorblind. Now, after that, we discussed about the hemophilia, which was the X-linked recessive trait. Okay. This is from where we have to start, right?
Yes.
Now, see, in the case of hemophilia, like normally in our body, there are like a lot of clotting factors present. Okay. Clotting factors, which are like important. For what purpose? It is important for blood coagulation. Like whenever we get a cut or if we get an injury, right? We don't bleed to death. Why? Because our blood clots, right? And for the clotting of blood, we have the clotting factors present in our blood. Okay, the clotting factors are there. Now, when a person has hemophilia, so that person has trouble in the blood clotting because they have a defective blood clotting factor. Do you know hemoglobin is written as HB? Now, the hemoglobin is made up of two chains, okay, alpha chain and beta chain. And accordingly, if the mutation is happening on the alpha chain, so the disease will be called as alpha. Wait. No, this is about the clotting factor. Sorry, I was telling you about thalassemia. See, so in the case of hemophilia, the person who has hemophilia suffers from this defective blood clotting factor. In this, again, the mutated gene is XH. H, which is like basically denoting that it is hemophilia that they are talking about. So XH gene is there, and the female again, the possibility of a female becoming hemophilic is very rare. Why? Because in order for a female to become hemophilic, the mother has to be a carrier, at least a carrier, and the father has to be hemophilic or affected. But remember, whether it is the male or whether it is the female, anyone, if they have hemophilia, will not stay alive for a long period of time. Because their body, because it's not possible that no one will have any kind of injury or any kind of cut ever in their lifetime, isn't it? That's not possible. Someone, like, even if a scratch is put, they'll keep on bleeding because their blood will fail to coagulate. Understood?
Okay. There's no cure for it?
No, because this is again a mutation. It's a mutation. It's okay. So, and
cannot be cured.
huh?
Mutation cannot be cured at all.
No, mutations can't be cured. There may be some preventive measures or medicines for this, but I don't think because it is like, see, if a person doesn't have something, then that thing could be added later on. If a person has excess of something, then that thing could be reduced later on. But if a person is born with absence of the gene only, or the gene is mutated, so there is no gene therapy as such for these kind of diseases. Understood?
Okay.
Also, in this case, mostly the hemophilic female would die much before even being born because of this problem. Clear? So, these are all the reasons because of which a female being hemophilic and being alive is very much difficult. So, this much is clear.
Yes.
After that, there comes this next disease, that is sickle cell anemia. Now, if you see, there's nowhere X written. Then what does that signify for us?
It's autosomal. No, autosomal.
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 Autosomal disease, right? Meaning it doesn't require the X chromosome for the inheritance or for anything of that sort. Now see, the sickle cell anemia is governed by the gene which is HB. Okay, now you tell me one thing. Which is the blood cell that is responsible for carrying the oxygen, I mean the gases in our body?
RBC.
RBCs. And why can they do that?
Maybe because of the surface area.
And this is one
And
of
they have red pigment. They have because of the hemoglobin is there, of course.
the main thing is hemoglobin, right? Hemoglobin carries the oxygen or the carbon dioxide. So what happens over here is that a person that has sickle cell anemia, they get this mutated gene HBS. Normal person has HBA, HBA gene. Okay, a person that is a carrier would have HBA, HBS. We don't know whether these are females or males. It could be anything because it's not X-linked. HBS is the diseased person and they die before attaining maturity. Again, now what happens in this disease? See, this is like the HBS is a mutated gene yet again. Okay, because all of these diseases are because of mutation. So it is a mutated gene. So which type of mutation actually occurs? Transversion mutation occurs. Now see, like I was telling you, the hemoglobin is made up of two types of chains, alpha and beta chain. Okay. So when this beta chain is synthesizing or it's being synthesized, so there is like a sequence and the things that are, you can say, present will be the amino acids, right? So because of this mutation, the hemoglobin's beta chain at the 6th position, it was supposed to have glutamic acid, which is the amino acid. But because of the mutation, the glutamic acid gets replaced by the valine amino acid at the 6th position. So this is what happens during the mutation. But what is the phenotypic result or what is the result that we see from this? Okay, what is the shape of RBC?
Biconcave.
Biconcave disc shape. And can you tell me one more important point about RBC which makes them very different from the rest of the cells?
They contain hemoglobin.
That's okay. In terms of its structure.
In terms of structure, they have a large surface area.
Why? Why do they have a large surface?
Because it is biconcave.
Okay. Any more reason? You are right, but I need one more thing which I am wanting to hear.
It is more a number. I don't know.
RBCs, they do not have a nucleus. They are enucleated cells. This is the very reason why they have more space to carry more gas molecules. Okay, they do not have a nucleus of their own. So, other than having a biconcave shape, that increases their surface area. The absence of the nucleus also gives them more area for the carrying of the gases. Now, what happens here? This is the normal shape of RBC. And because of that transversion mutation that took place, the RBC changes and polymerizes from this disc-like shape into sickle-like shape, like this. The RBCs become like this. Now, do you see what's the problem with it?
Space reduced, the area reduced.
Space reduced. That means less gases or less oxygen will be delivered to the body. And if our body, if our organs do not get the right amount of oxygen, what will happen? We'll die. Isn't it?
Yes.
Yes. So that is why, like, this is what the disease is. So the mutant hemoglobin, it undergoes polymerization under low oxygen tension, causing the change in the shape of RBC from biconcave disc shape to elongated sickle-like structure. This is what sickle cell anemia is. Okay, the RBC becomes sickle cell instead of being a biconcave disc shape. Understood?
Yes.
After that, this one you've already studied a little bit, phenylketonuria in the pleiotropy. Now, this is the error of the metabolism. Okay, Now this is yet again autosomal recessive kind of disorder. Here, normally in normal individual, there is an enzyme which is important, and that enzyme is called as phenylalanine hydroxylase. Okay. Now this enzyme is important in our body. Why? Because this enzyme converts the amino acid called as the phenylalanine into tyrosine. Okay. Now, if this enzyme will not be present, it means that the phenylalanine will not be getting converted into tyrosine, isn't it? So, as a result, the accumulation of phenylalanine starts to take place. Now, once the phenylalanine gets accumulated a lot, it gets converted into phenylpyruvic acid because of that accumulation.
Okay.
And the accumulation of phenylpyruvic acid now causes all of the disease or all of the phenotypic expressions that we saw: mental retardation, reduction in the hair, skin pigmentation. All of these problems come along because the absence of the enzyme phenylalanine hydroxylase, it itself is an abnormality, a disease. Other than that absence, there are three other things that happen. That is why we took this as an example of pleiotropy. Understood?
Yes.
After that, there comes thalassemia. Now, thalassemia is yet again autosomal recessive disorder. This is caused by mutation or deletion, particularly, meaning a gene is getting deleted because of mutation. So what here, what basically happens over here? Again, defective hemoglobin is made, abnormal hemoglobin. Okay. Now you know already that the hemoglobin has two chains, alpha and beta. So if the alpha chain is affected, then the person would have alpha thalassemia. If the beta chain is affected, so the person will have beta thalassemia. Clear?
Yes.
So here, in the case of alpha thalassemia, the production of alpha globin chain is affected, whereas the beta thalassemia, the beta globin chain is affected. Now, HbA1 and HbA2 genes, these are the genes that actually get deleted. Okay, and where are they present? They are present on the chromosome number 16. So basically, the chromosome number 16 is governing the formation of alpha chain. And you know that the chromosomes, they carry the genes. So particularly HbA1 and HbA2 gene, if they get deleted, the person has alpha thalassemia. Clear?
Yes.
Same way, for beta thalassemia, HBB gene is present on the chromosome number 11. So if that is deleted, then the person will have beta thalassemia. Understood?
Well, what is it here? HBB gene.
This is the name of the gene. Here HBB and here HbA, HB2. HbA1 and HbA. Clear?
Okay. Yes.
Now let us see the comparative analysis of thalassemia and sickle cell anemia. Why have I added it? Because I have seen a question that compare how thalassemia and sickle cell anemia are different from each other. So firstly, thalassemia is a qualitative problem. Qualitative means the quality of the hemoglobin can either miss the alpha or beta chain. Because of that, we call it as the qualitative problem. Okay, where too few globin molecules are being synthesized. But if you think about sickle cell anemia, wait, I just said the opposite thing. Thalassemia is a quantitative problem, quantity, because less amount of globin chains are being synthesized because of the mutation, because deletion took place. Whereas sickle cell anemia is a qualitative problem wherein the quality of the RBC is changing, or the hemoglobin is sort of getting reduced. Understood?
Okay.
So this is the major difference between thalassemia and sickle cell anemia, that the thalassemia is a quantitative problem, sickle cell anemia is a qualitative. Any doubts?
No.
Now see, genomatic mutation. Now, just like how we saw the gene mutation, which was basically point mutation that we studied, now the mutation that happens in the chromosomes, because of which the phenotypic expression changes, that is what we call as genomatic mutation. It is of two types, aneuploidy and euploidy. Okay. Now see, aneuploidy is When the two and the diploid situation has one less, so you call it as monosomy. If it has two less, then you call it as nullisomy. And if it has one extra chromosome apart from the diploid chromosome number, then you call it as trisomy. So monosomy, nullisomy, and trisomy are the examples of aneuploidy. Is it clear?
Okay.
After that, there is the euploidy. Basically, euploidy is like increase in the sets. Okay. So like how when we talk about the sets of chromosomes, so haploidy is like one set of chromosome. Polyploidy is more than two sets, like even more than diploid is polyploidy. Exactly three sets of chromosomes would be triploid. Understood? And polyploidy is also like a unique situation. Understood?
Okay.
So basically, like if I would compare euploidy and aneuploidy, so what would be the difference? That in the case of euploidy, we are concerned with the sets. Either the increase in the set or the decrease in the sets of chromosomes. Understood? Whereas the aneuploidy will be just no set, like not concerned with set of chromosomes. Getting my point?
Yes, ma'am.
So not concerned with sets of chromosome. So it's like one increment, two increment like that, or decrease like that. But no sets are involved over here. But in the case of euploidy, sets are involved. 3 set, 2 set, 1 set like that. Clear?
Yes.
Now we come on to this topic, that is chromosomal disorder. Now this topic doesn't revolve around mutation. Rather, it is like these diseases are because of the chromosome number imbalance. Like there is an imbalance in the number of chromosomes because you remember in yesterday's class I told you because of faulty fertilization.
Sorry, I can't. Sorry, again, please.
I said, do you remember how I explained that the chromosomal disorder would be mainly because of the faulty fertilization, like abnormal sperm fused with the normal egg or abnormal egg fused with the normal sperm, like that. So the first disease under that is the Down syndrome. Now this disease was first described by Langdon Down in the year 1866. And this is the trisomy of the chromosome number 21. Meaning at the chromosome number 21, there is the presence of an extra chromosome. Understood?
Okay.
So instead of a person, like a person normally has 46 chromosome, right? We write 45 plus XY, which means that either the X or the Y. So this is like the diploid number. 46 chromosomes are there. But here, the person with Down syndrome, they have 47 numbers in the chromosome. They have one extra.
Okay.
So this one, it is characterized by, like now the person that has Down syndrome, that person would have a short stature, meaning they would be of short height. They'd have a small round head, furrowed tongue. Their tongues are always protruding out of their mouth a little bit. The palm, like how we have the normal lines in our palm, they have a lot of creases in our palm, a lot of lines, more than us. They have a big wrinkled tongue. Now their physical, psychomotor and mental you can say structures and the capabilities are underdeveloped. Their gonads and genitals or the genital area, they are also underdeveloped. Clear?
So
And
not reproduce. They cannot reproduce.
yeah, they can or they cannot. Like either of the case. If like there are many people that undergo corrective surgeries and then they can reproduce. Okay. Because but mostly it's like majority of them, they are underdeveloped in terms of many functions, like mentally also they are underdeveloped, physically also they are underdeveloped, like that. And they have like almond shaped eyes that are a little bit slanted. So that is the Down syndrome. Then comes Klinefelter syndrome. Now Klinefelter syndrome, it affects only the males, and it's like normally a male is supposed to have one X chromosome and one Y chromosome. But the people with Klinefelter syndrome, they have one extra X chromosome. So they have XXY situation.
Okay.
Instead of being XY, they are XXY. So this
In
defect.
the previous one, what was extra, X or Y?
Which one?
Uh,
Here. No,
yes.
one chromosome number 21 was extra. This is not X or Y. This thing,
Okay.
autosome was extra.
Understood.
But here the sex chromosome, X chromosome is extra. So this defect would occur due to the union or the fusion of the abnormal egg, which has two X chromosomes. Normally, an egg is supposed to have one of the X chromosome because egg is haploid. And when that will fuse with the normal sperm, then we call it as, like, this is how the defect might arise. Or the normal egg is there, which has just one normal X chromosome, and then the abnormal sperm, which has X and Y. So now these people, they are sterile males. Now they cannot reproduce. They have overall masculine development. They would look like males, but because of the presence of the extra X chromosome, they would be like also consisting of some feminine characteristics. Like they would have a feminine pitched voice. They'll have development of breast, which is called as gynecomastia. Clear?
So
Not,
they're trans?
no, they're not trans. No, they're not trans. So it's like they have this extra X chromosome because of which they have, like, they are sterile and they possess all of these properties. Understood?
Yes.
The last disease that comes under the chromosomal disorder is the Turner syndrome. Now, this one affects only the females, and this is because of monosomy. Monosomy means 2n-1, that is one chromosome is missing. Like either the X would be missing or the Y would be missing, like that. So it appears because of the fusion of the abnormal egg that has the autosomes but doesn't have any X chromosome and a normal sperm. Or it happens in the case wherein the normal egg is there but the abnormal sperm is there. So these females, they, because of the absence of XX, they are sterile. Why? Because their ovaries are rudimentary. They're not very well developed. They're very poorly developed. That is why they're sterile, because they cannot further reproduce. And they also lack secondary sexual characters, meaning they do not have their menstrual cycle. They do not have female like body structure or puberty, like how female develops their breast, and um, all of the secondary sexual characters like the broadening of the waist. These things do not happen for them because they don't have the menstrual cycle. Understood?
Yes.
Okay. Now tell me, what is the last thing you've written? Pleiotropy.
Yeah, completely clear topic. Chapter done?
Hmm?
Chapter done, ma'am?
Only one topic is left and the chapter is done. After this, which chapter shall we be starting? Molecular basis.
Yes.
See, before doing molecular basis class, I want you to come prepared with 2 to 3 topics, I'll tell you. Okay, because uh, yeah, when you'll come prepared, then you'll not have any problem. Because
Okay.
what about your that chapter from 11? How is your biomolecules?
In
From
11th,
class?
we had biomolecules.
Huh?
In 11th, we had biomolecules.
11th.
11th class. Did we have biomolecules?
Yeah, you
Yeah.
did. I can understand. Okay. So you'd have to brush up a little bit. Okay, so I'll tell you. Oh wait, I'll not tell you. I'll teach you. Don't worry. Okay, you know that. You know that.
From here?
Okay.
Sorry. Yes, ma'am. Yes, ma'am. The disorder. Yes. Yes, ma'am. Yes, ma'am. Yes, ma'am. Color blindness. Ma'am, crisscross inheritance. Color blindness next. Yes, ma'am. 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 Yes, ma'am.